Welcome to Life in Biology. I'm Dr. Joel Graff. Today we're going to continue our discussion about CRISPR, and what we're going to be talking about is gene drives and how they defeat Punnett squares. Now in episode 3 I talked about a famous scientist named Gregor Mendel. Mendel did these experiments with pea plants where he followed 7 different traits, and he followed the traits from one generation to the next and kind of figured out the mathematics of how genes are passed on. So in a nutshell, Mendelian genetics suggests these things. Traits are inheritable, so if you look at what the parents' traits are, you can guess at what ratio of those traits will be passed on to the next generation. Traits are determined by genes. Mendel didn't use these terms, but that's how you talk about Mendelian genetics, is through genes. Now, one thing that's interesting about biology is you have 2 parents, and each parent gives you a set of chromosomes. On each chromosome that you get from one parent or another, there's usually 2 genes per locus. Locus is a specific position within a chromosome. So chromosome 1 position whatever, you get that gene from your dad, and then chromosome 1, that same position, you get a gene from your mom on that second chromosome. So you get 1 gene from your mother and 1 gene from your father. There's this idea of dominant and recessive genes. So sometimes you can have a 50-50 mix of genes where you've got, say, brown eye color gene versus blue eye color gene, and if that happens, you're going to always get the brown eye color coming out. So in that case, brown is dominant, so you need to have a blue gene from each of your parents if you want to have blue eyes. And there's basically some pretty simple math that goes into predicting the traits of the offspring. That's assuming whatever trait you're looking at is a 1 gene trait. There are plenty of traits that take input of multiple genes, and so then the math gets a little bit more difficult. But anyway, if you're talking about let's just say this is one of your parents, and let's say this is eye color, you typically get 50-50 inheritance from heterozygote parents. So here are 2 different chromosomes, and the gene we're looking at is here in the non-red colors. And what this happens here is this is a heterozygote, so you've got, let's say, a brown eye gene color and a blue eye gene color. Now if you look at the offspring, 50% of the kids from this parent are going to get the blue eye color, and 50% are going to get the brown eye color. So that's how it typically works. So when you're talking about mixing 2 different parents together and predicting what their offspring are going to have, a good way to do that is called a Punnett Square. So this is taking Mendel's math and just putting it in an easy to digest format. So let's say parent 1 has 2 different traits, and here we're going to say little r means no resistance, let's say just to infection with a certain thing, and then capital R is going to be resistance to infection with a certain thing. And in the case I'm thinking of is, let's say instead of people, we're talking about mosquitoes, and resistance would be resistance to being infected with a malaria parasite. So you have one parent that has both little r, so it has no resistance to the parasite. The other parent has 1 gene that is a capital R that provides resistance, and then 1 gene that's a little r that doesn't provide resistance. So 50% of your offspring are going to be capital R, little r. And by this, I'm taking this chromosome from this gene from this parent, mixing it with this gene from this parent. Over here, I'm taking this. For this square, it's this gene from this parent and this gene from this parent. And then down here I've got the small r from parent 1 and small r from parent 2, and same thing, small r from parent 1 and small r from parent 2. So 50% of the offspring are going to have the capital R, little r feature, and if capital R is a dominant gene, that means 50% of the offspring are going to have resistance to malaria, and 50% of the offspring are not going to have resistance to malaria. Now, to mix things up, gene drives are selfish. So let's say that you've got here's your we'll call it your resistance gene in the green. Now the resistance gene on one chromosome, and then the no resistance gene on the other chromosome. But what if we also include a guide RNA and Cas9 in this on this chromosome right next to this gene? What we can do is then we can have the Cas9 and the guide RNA go and cause a double-stranded break right there where the blue arrow is, and what's going to happen is it'll cause a break there, and then you get homology-directed repair where this black gene is then fixed by having the resistance gene in there along with the Cas9 and the guide RNA in both strands of your chromosome. So what gene drives do is they turn heterozygotes into homozygotes. Now, what that means is that all the offspring from this are going to get this chromosome or this chromosome, and since it's homozygous, 100% get this red, blue, green, purple, red stretch of DNA. So 100%, where we had 50-50 before, if this hadn't attacked that and it was heterozygote to begin with, you'd typically get 50% this, 50% that. But because it's able to knock out the gene in the second chromosome, you get 100% homozygous. So that changes your Punnett Square a lot, because if this was parent 1 and this is parent 2, the genes that it would pass on, parent 2 is always going to be homozygous for resistance. Parent 1, this might be a mosquito out in the wild. When these cross, you're going to get capital R, small r, but remember the small r is susceptible to the gene drive because of the CRISPR, and the small r is going to be converted to a capital R through the recombinant directed repair. That's going to happen in this offspring, this offspring, this offspring, and this offspring. So no matter what way you cross these parents, you always would have ended up with a heterozygote, but now you're ending up with always a homozygous for the resistance, and so 100% rather than 50% will show that trait. So what does this all mean? Well, what this means is that typical Mendelian genetics, if you have a heterozygote, like you release a mosquito that has a resistance to malaria, it's got the resistance on one gene and not the other, and then you've got your wild type population. And so here resistance is just the green. If you see green in it's going to be resistance. So you cross your lab made mosquito with the native mosquito, you're going to get, half the time you're going to get that one chromosome, the resistance gene passed on, but the other half of the time you're going to get that resistance gene not passed on. And these are going to be crossing with the natural population, which is always the black circle. So after this first generation, you have a 50-50 mix of offspring that have the resistance. But then when you go through the math and look at how things happen, the next generation, only 25% of your offspring are going to be this. So or have the resistance. So the other 3/4 are going to have the double circle, the double lowercase r, and it's because we're outcrossing into a population that doesn't have the resistance, and so you just end up with having whatever gene, the resistance gene, for example, weeded out. Now in a gene drive, instead of being half a circle filled, we've got the full circle filled, and it crosses with a native mosquito that doesn't have the resistance. In the first generation, this mosquito is an offspring and this mosquito is an offspring, and they started out as heterozygotes, but then they turned to homozygotes because of the gene drive. Now they outcross with the native population, and you have the 4 possibilities you can get. You have 2 genes here, 2 genes here. You're going to end up with a heterozygote, but that heterozygote is going to immediately turn to a homozygote because of the gene drive, and 100% of your next generation is going to have that resistance